Hey everybody. This recording is going to be about capillary exchange in the cardiovascular system. And again, here we're looking specifically at blood capillaries, but some of the things that we talk about here are going to be applicable to lymphatic capillaries as well. Now, this is a close-up of a fenestrated capillary where there are a couple of things I want to remind you of, particularly if we look at the structure of it up here. We see that there are these endothelial cells with a thin basement membrane on the outside. And again, it's only in this thinnest part of the circulatory system are materials able to get exchanged. Many places along the ways will have these tight junctions between adjacent endothelial cells. And the thing about tight junctions is they're kind of a permeability barrier, so they will prevent most materials from getting outside. Now, if we were to zoom in on this this lower part here of the capillary wall, there are a few main ways we see blood materials leaving the bloodstream. Things that are common across all types of capillaries are, first of all, we see just plain diffusion for things that are lipid soluble. So just like steroid hormones were able to sink through the cell membrane, lipid soluble substances are able to do the same, so they're able to pass through the capillaries without much interference. There is some movement, but some relatively limited movement of water soluble materials between the endothelial cells as well, but primarily in places where you don't have those tight junctions. In places where you do have tight junctions, that is largely it's slowed down and kind of minimized, I guess it's fair to say. Another place that we see large materials or things that basically lots of substances will get passed from one side to the other through through these sort of this penocytosis process where little bits of plasma will get ingested here into these vesicles and then dumped out on the interstitial fluid side. And then last, the other way the materials can pass through is through the fenestrations. Now, as you'd expect, this would only be true for fenestrated capillaries, although larger materials can also pass through the sinusoidal capillaries too. But in the case of the continuous capillaries, those most common capillaries in the circulatory system, it's really only, methods 1, 3, 2, and 4 that we see present there. Just diffusion, active transport, or movement in places where you don't have tight junctions. But anyway, those are all the ways that materials are going to be moving from one side of basically from the lumen of the capillary into the interstitial spaces. Now, there's actually a pretty helpful figure in your text that describes how capillary exchange happens, and we're going to kind of work this out kind of bit by bit. The first thing to talk about is the kinds of pressures that are present that are kind of all together construct the local blood pressure. Now, the net amount of fluid that leaves the blood capillary is determined by the pressure that's called the net filtration pressure. Now, I think we talked about filtration in class, thinking about material stuff kind of getting strained out, say, through the capillary bed. Basically, there are two kinds of forces that oppose one another. The first kind of pressure is what we call hydrostatic pressure, and this sort of like when we talk about blood pressure overall, this is the pressure of the fluid against the capillary wall. So this is like the pressure from the weight of the water, basically. So this hydrostatic pressure pushes fluid out of the capillary. That's the net effect of it. On the other hand, we know that there are lots of plasma proteins, and those plasma proteins have a certain attractive force for water as well. So osmotic pressure works in opposition to hydrostatic pressure. basically these solutes aren't able to diffuse out of the capillary by themselves, so they tend to suck the fluid back in. Okay, so hydrostatic pressure pushes fluid out, and so it allows filtration, and osmotic pressure does the opposite. It sucks fluid back in. So when we calculate the net filtration pressure, this is done by summing up all of these different forces. For this panel, we're looking at the alveolar side of the capillary bed. So, just to back up, when I say the alveolar side, I mean this part here that's closer to where the alveolar. I was going to say arteriolar side. The arterial, the smallest vessels that are going to be sending blood to these capillary beds, assuming the capillary sphincters are open, there's generally fluid is leaving the bloodstream on this side, but it's going to tend to get sucked in on the other side. So just to go on with this here, now the width of these arrows you can consider to basically they're kind of scaled relative to one another. So on the arteriolar side of a capillary, you see that there's a hydrostatic pressure of about 35 millimeters of mercury. So that's the fluid that's pushing the fluid out from the capillary lumen into the interstitial fluid. At the same time, the dissolved solutes inside the plasma that can't leave the plasma have a osmotic pressure of 26 millimeters of mercury. And then, but that's just what's happening from the from the capillary's point of view. On the interstitial fluid side, the side where the cells are, there's a small amount of hydrostatic pressure that's kind of, here they call it zero, but they look like it's going in. There's a very almost zero pressure pushing fluid back into the capillary, but there's some sucking force because of the amount of fluid that's being pulled out. So, I mean, you can add things like this if you like. This is showing, so the net filtration pressure calculated as the sum of the capillary's hydrostatic pressure plus the osmotic pressure of the interstitial fluid. You can see hydrostatic pressure is pushing it out, and the osmotic pressure of the interstitial fluid is also sucking it out, minus the interstitial fluid hydrostatic pressure, this part that's going to tend to try to push it in, plus the osmotic pressure of the capillary, where it's getting sucked in. When I do this, I usually pick one side. I usually pick the capillary side, and I'll say like it's a plus 35 out or a negative 26 out, kind of for sucking in, you can, do the signs any way that you want. But these are like decent general values for looking at capillary exchange. But anyway, the net effect here, when these are all summed up, is there's a net filtration pressure of 10 millimeters of mercury out. Right? Meaning, on the arteriolar side of the capillary bed, fluid is getting squeezed out. Over the course of the day, about 20 liters of fluid altogether is going to leave the bloodstream, eventually to get sucked back in one way or another. Now, on the venular side, on the other side, there are some differences. So I'm just going to kind of tap back and forth here a little bit. So notice first the hydrostatic pressure of the capillary. This is again the pushing force of the fluid leaving the capillary. Here it's 17 millimeters of mercury. On the other side, on the arteriolar side, it's up at 35 millimeters of mercury. So you notice it's decreased substantially by the time it gets to the other side. Now here, the osmotic pressure, this 26 millimeters of mercury, again, that's the sucking force of those plasma proteins in. And you'll notice it is exactly the same as it is on the other, right? So those plasma proteins, by and large, aren't leaving the plasma. They are assisting with this kind of water balance. Then again, 0 millimeters of mercury for the hydrostatic pressure of the interstitial fluid, and 1 millimeter of mercury for the osmotic pressure of the interstitial fluid. To kind of flip back again, those pressures are the same. Okay, so then what's the difference between these two panels? It's only the hydrostatic pressure, which is much lower on the venular side than on the arteriolar side. Now, so then the net effect of this is it's a net negative in. So basically, this is going to be a net sucking in of about 8 millimeters of mercury, as opposed to a net out of 10 millimeters of mercury over here. Now, in case you're thinking, hey, that doesn't add up, it doesn't look like it's balanced, you are correct. So if you go back here again, I mentioned before, we see 20 liters of fluid leaving the capillary beds over the course of a day, but only 17 of those liters is getting reabsorbed on the venular side. Now, that extra fluid that's there would stay marooned in the tissue beds if it were not for the lymphatic capillaries that were also there. Lymphatic capillaries, you'll see in the next chapter, they're kind of structured like blood capillaries are, but they're made to be like more absorbent in a way. The endothelial cells don't overlay one another as easily, so they're really absorptive, right? Well, that's one of the main things that the lymphatic system does for us, is it prevents that like 3 liters of fluid per day from waterlogging your tissues as edema, which is spelled edema. Without the lymphatic capillaries, we'd have edema particularly in the lower extremities, and it would also have the effect of decreasing our blood pressure. You figure if we were losing that amount of fluid, 3 liters of fluid, that's a substantial part of your total plasma volume. So anyway, we're going to talk more about what the lymphatic capillaries do in class and in another video. But for right now, understand that it's not totally balanced, fluid leaving and fluid entering the capillary bed. But that was actually all I wanted to say. The big thing to get about this is that people have tended to get hung up on this, as I've taught this in previous years. The main thing I want you to take from this is certainly the definitions of the pressures. With that, hydrostatic pressure pushes out, osmotic pressure sucks back in to the capillary or the interstitial fluid, and really the pressure that is changing is the hydrostatic pressure is higher on the arteriolar side and lower on the venular side. So in the case of when the blood is closer to the arteriolar side, you see a net out of fluid leaving the capillary bed, and then on the venular side, a net in, where wastes, particularly wastes, if you want to think about it that way, but also hormones and foods and other stuff like that are going to be entering the bloodstream there. Anyway, that's the main stuff. I will see you in class.